Uh, to start with today's class, I want to revise what all we discussed in our last class. I don't know whether all of you have attended to my last class or not, but uh, I want to revise the things what all we have discussed in yesterday's class. In yesterday's class, we have discussed about the importance of some of the molecules which are acting as an insecticide. And along with the introduction of these molecules, we have studied about the structure, their toxicity, and the mode of action, and the metabolism. We have covered these things for few of the molecules like neonicotinoids, nerestoxin, spinosad, and ivermectins. Under ivermectins, we have studied about abamectin and ivermectin. These things we have covered in our yesterday's class. Okay, just to have a glance, I wanna give you the summary of these things. Then after we'll enter into the rest of the molecules which we have to study under unit 10. Okay, and coming to the nerestoxin which we have studied yesterday. It is a naturally occurring poison isolated from marine annelid, lumbrineris, that is spinosad. It is acting as an active ingredient and was obtained from soil actinomycetes, and it's a mixture of two complex molecules, spinosyn A and spinosyn D. Neonicotinoid, and it is a systemic and is extremely effective against certain pests. They interact with the acetylcholine binding site of the nicotinic acetylcholine receptor and acts as agonist. This is what we discussed yesterday uh, in our class along with the ivermectin. Here, ivermectin acts as agonist for gamma aminobutyric acid, that is GABA dated chloride. In the treatment of different diseases caused by nematodes in the veterinary field. This is what we discussed. And coming to the today's topic, milbemycins. Before
Ma'am, your voice is not audible.
in your lower classes about the data channels, about in cell signaling or cell biology. Coming to the data channels, these are specialized proteins present on the cell membranes, which controls the flow of ions across the membranes in response to specific drug or ligand or any alkaloid. And coming to the things what we are discussing every day while we are talking about different molecules which are acting as an insecticides, we'll talk about voltage gated channels. What are voltage gated channels? These are the channels that will open or close in response to the electrical potential which is present on the cell membrane. Okay, and here Yesterday also we have discussed about the importance of nicotinate acetylcholine receptor, which comes under ligand-gated channel. These channels are also open and close in response to the binding of a specific chemical ligand. Example, any molecule, nicotinate acid, or because why we are talking that it as nicotinate acid, it is acting as a neurotransmitter to the channel or an associated receptor. Coming to the nicotinate acetylcholine receptor, usually it is present at neuromuscular junctions, which is a part of the central nervous system. And these channels open when acetylcholine binds to them and allowing sodium and potassium ions across the membrane, which mediates the synaptic transmission. And coming to the GABA receptor, that is gamma aminobutyric acid receptor, which is present in the brain where the chloride ion channels open when the GABA dosan binds to this receptor, leading to hyperpolarization. No, I am not turning any slide here. Just I am giving you the revision about the things what we discussed yesterday. Just a summary. Leading to hyperpolarization of the cell membrane and inhibition of neuronal activity. We have studied about these two receptors yesterday while we are covering neristoxin, avermectin, neonicotinoids, abamectin, etc. Okay, we have studied about these things, and today we are going to talk about more or less you are going to see the same type of ligand gated channels or voltage gated channels in all these molecules, where this molecule acting as a chemical and binds to these receptors and which is stimulating the cell in order to act accordingly. Today our topic is milbemycin. These are a group of macrocyclic lactone compounds. In our previous class also, we have studied about these macrocyclic lactone compounds, where you have seen avermectin is a type of macrocyclic lactone compound. Slightly, there is a variation in the structure, but more or less all are same. These milbemycins are a group of macrocyclic lactone compounds that are used primarily as antiparasitic agents. And these are derived from bacterium Streptomyces hygroscopitus, subspecies Arylotrematosus, and milbemycins are chemically related to avermectins. Yesterday, we have studied about the avermectins, where it is also acting as an insecticide, or it is acting as one of the antiparasitic agent. Okay, and coming to the avermectin, it is if you could see the structure of this avermectin, it contains two different molecules in that. It's a complex compound where it contains avermectin B1A and avermectin B1B. These are the two things which you are going to see in avermectin. Yesterday, even you have seen the structure of avermectin also. So more or less, milbemycins are chemically related to avermectins. These are another class of macrocyclic lactones. And if you could see the structure of avermectin, it contains lactone rings. If you could see here, these are the lactone rings which are important for the biological activity. And a total of 16 lactone rings are present. That is, it is 16-membered lactone ring. If you could see the structure, it is similar to that of avermectins, but milbemycins lack the disaccharide group which you have seen in avermectin. This is the structure of milbemycin, which contains ketone group and hydroxylate group, etc. Coming to the mode of action, these milbemycins exert antiparasitic effect through interactions with specific ion channels in the nervous system of parasites, which leads to death and eventually, which leads to paralysis and eventually death of the organism. And coming to the channels, how it shows its interaction. The first one is glutamate dated fluoride channel. As an introduction, I have given you the idea about the dated channels, where ligand comes and binds to the receptor to activate the cell. Okay, and here now we are going to talk about that is glutamate dated chloride channels. The first one here is milbemycin that will go and bind to the glutamate dated chloride channels, which is present on the cell membrane of invertebrates. And when the glutamate binds to this when the milbemycin binds to the glutamate dated chloride channel, this increases the permeability of the cell membrane to the chloride channels and cause the influx of chloride ions into the cells. And the increased chloride ion influx leads to hyperpolarization of the nerve and muscle cells and making it too difficult for the cells to generate action potentials. If there is a problem with generating action potentials, as we already know, these dated channels involves in physiological activity. So when this DAT fails, obviously it leads to the alteration in the physiological activities or the cellular functions, or failing in regulating the cellular functions that leads to the death of the organism. And the second one is gamma aminobutyric acid dated chloride channels. As an introduction, we have studied about this GABA receptor under ligand dated channels. These GABA receptors are present in the brain, and the chloride channels open when the GABA comes and binds to the GABA receptor, okay, which leads to hyperpolarization of the cell membrane and the inhibition of neuronal activity. So here, when the milbemycin goes and binds to GABA-dated chloride channel, these shows inhibition towards neurotransmitter receptors, contributing to the paralysis of the parasites. Sometimes eventually it leads to the death of the organism too. So we already studied the effect of these things. The first case is paralysis and the second one is the death. Okay. And here, the next one here is, if you could see the milbemycin, under milbemycin, we have studied about two things, structure and the mode of action. Coming to the milbemycin structure, it is a 16-membered macrocyclate lactones with various functional groups. And coming to the mode of action of milbemycin, they target GABA-dated chloride channels in pests or parasites, and that cause hyperpolarization of the cell membranes, leading to the death of the parasites and eventually, leading to paralysis and the death of the parasites eventually. This is what we discussed about the milbemycin. And coming to the next topic, fipronil. This fipronil is a broad spectrum phenylpyrazole insecticide, which is used to control various pests. Okay, and it is widely used in agriculture and veterinary areas to control a variety of insect pests, and it is effective against insects like fleas, ticks, ants, cockroaches, etc. And it works by targeting the central nervous system of insects, leading to the death. Coming to the structure of this, if you could see the structure here, it has a phenyl ring with dichloro and trifluoromethyl groups and a pyrazole ring with several substituents. Okay, and the chemical name of this fipronil is 5-amino-1,4-H pyrazole 3-carbonitrile, and the molecular structure consists of phenyl ring and pyrazole ring. This is the chemical formula. It has 12 carbons, 4 hydrogens, and 2 chlorines, F6 and 4 oxygen sulfur, and the molecular weight is 437.15. And coming to the physical and chemical properties, it is white crystalline solid whose melting point is 200 to 201 degrees Celsius, and it shows low water solubility but soluble in organic solvents, and it is stable under normal environmental conditions, but it gets degraded when it gets exposed to the sunlight for a longer period. And coming to the mode of action, here this fipronil targets GABA as well as glutamate-gated chloride channels that will cause hyperexcitation and leads to the death of the insects. This is about the mode of action. If you could see in detail here, fipronil disrupts the central nervous system of insects by 2 channels. The first one is GABA-gated chloride channel and the second one is glutamate-gated chloride channels. This fipronil molecule goes and binds to the GABA-gated chloride channels in the neurons and blocks the channels so that it can prevent the chloride ions entering from the nerve cells. And when it gets inhibited, that leads to excessive neuronal stimulation because the neurons cannot repolarize properly, which results in hyperexcitation. And coming to the next one, glutamate-gated chloride channel. Here, this fipronil affects glutamate-gated chloride channels and enhances its neurotoxic effects. And the blockage of these channels contributes to uncontrolled nerve impulses, leading to hyperexcitation of the neurons that leads to uncontrolled muscle activity, convulsions, paralysis, and death of the insect. This is how this fipronil acts on 2 different gated channels, leads to the alteration in the nerve impulses, causes paralysis and the death of the insect. Coming to the metabolism of the fipronil, if you could see here, in different ways we are seeing here, this fipronil undergoes several metabolic processes in insects, animals, and the environment. Here, this fipronil metabolized in animals and insects primarily to fipronil sulfone with further degradation in the environment. And here, if you could see this fipronil metabolism in animals, this is primarily metabolized in the liver by an enzyme called cytochrome P450 and forms fipronil sulfone, which is a major metabolite. There are other metabolites that include fipronil disulfinyl, fipronil sulfide, and various conjugated forms. And metabolism in insects, there are several metabolic pathways which are similar with fipronil sulfone. Being a significant metabolite, the metabolites retain some level of insecticidal activity and contributes to fipronil's effectiveness. Whereas in case of the environment, this fipronil can degrade under sunlight through photolysis to form fipronil disulfinyl, which is more persistent and less toxic. In soil, the fipronil can bind to organic matter and undergo microbial degradation. And in water, it has a low tendency to leach and tends to remain bound to sediment. Coming to the hydrolysis of fipronil, hydrolysis means the breaking down of a compound due to reaction with water. For fipronil, this process is influenced by environmental conditions such as pH, temperature, and other chemicals. Here, if you could see the hydrolysis of fipronil, under the hydrolysis of fipronil, the first one is hydrolysis rate. This is relatively stable. Fipronil is relatively stable in neutral to acidic conditions, but can undergo hydrolysis more rapidly in alkaline conditions. However, under alkaline conditions, degradation increases with pH, and the hydrolysis may not be the major degradative pathway for fipronil since it is stable at typical environmental pH. And coming to the hydrolysis products, one of the primary hydrolysis products formed when fipronil reacts with water is fipronil amide. Another product formed under certain conditions means in the presence of different chemicals and the environmental conditions, it forms fipronil sulfonate acid. And coming to the environmental impact, this fipronil undergoes hydrolysis that reduces the concentration of active fipronil in the environment, which potentially decreases in its long-term impact. However, the hydrolysis products themselves need to be evaluated for their ecological and toxicological effects. Coming to the antagonistic toxicity of fipronil. This fipronil toxicity was antagonized by, you can see a chemical here. It is not mentioned maybe, but there is one chemical called tributyl phosphoornithioate and piperonyl butoxide. It was antagonized by these two chemicals on the cockroach, and it was about 2.3 to 3 times less toxic to cockroaches pre-treated with the piperonyl butoxide. Coming to the other effects, apart from that, this shows its toxic effect with by interacting with other pesticides. The first one is fipronil can be used in combination with other pesticides, and these combinations can have antagonistic, additive, and synergistic effects. And coming to the mechanisms of antagonism, it shows competitive binding, metabolic interactions, and protective effects. And if you could see the environmental and health implications, which understanding this toxicity is important for risk assessment and developing safer pesticide use strategies. It can also help in designing pesticide formulations to minimize harm to non-target organisms. Those are beneficial organisms. By this, we completed fipronil. Here, under fipronil, we have covered about the introduction, structure, properties, mode of action, metabolism, and then hydrolysis of fipronil and antagonistic toxicity. Coming to the introduction, it's a broad spectrum phenyl pyrazole insecticide used to control different pests. And coming to the structure, it has a phenyl ring with dichloro and trifluoromethyl groups and a pyrazole ring with several substituents. And coming to the properties, it is white crystalline solid and it is low soluble in water and it is stable under normal conditions. And coming to the next part, mode of action, it targets two channels. The first one is glutamate-dated chloride channel and GABA-dated chloride channels, which causes hyperexcitation and causes the death in insects. Coming to the metabolism, it is metabolized in insects and animals, primarily to fipronil sulfone with further degradation in the environment. And the next part is hydrolysis of fipronil. Under hydrolysis of fipronil, how the hydrolysis occurs and what all the things which gets produced with the environmental conditions. We have studied all this. And we even covered the importance of it, where fipronil is more stable and neutral to acidic conditions or environment, but hydrolyzes faster in alkaline conditions, which produces the fipronillamide and fipronil sulfonate acid. And the last one we have covered is antagonistic toxicity, where this fipronil involves interactions in a combined effect of fipronil and other substance, which is lesser than their individual effects. And here you have seen the competitive binding, metabolic interactions, and protective effects impacting environmental and health risk assessments. By this, we have completed the fipronil part. The first one we have covered the milbemycins, and the second one we have studied the fipronil. And the third one we are going to talk about formamidins. Under the formamidins,
Ma'am, excuse me, ma'am.
Amma,
Could you please elaborate on the PBO as a synergist mechanism?
PBO, piperonyl butoxide.
Yeah, could you please elaborate the mechanism as a synergist?
How exact antagonist, right? Hello?
Yes, ma'am. Yes, ma'am.
Okay. See, the fipronil toxicity was antagonized. Here,
Ma'am,
first
you are
we'll
not
see.
audible.
Hello? Am I
Yeah,
audible?
now you are audible, ma'am. Yeah, now you
Yes.
are audible, ma'am.
Yes. This PBO actually it comes under fipronil, which is a semi-synthetic derivative actually. It is used as synergist in insecticides, and it acts as a antagonist by inhibiting the activity of the enzyme. We have studied one enzyme. The enzyme is cytochrome 450 enzyme. It is somewhere I have written.
E450.
Yes, cytochrome. Yes, here. Okay. It inhibits the activity of cytochrome 450 enzyme in the insects, which are responsible for detoxifying the insecticide within the insect's body. And by inhibiting these enzymes, this piperonyl butoxide prevents the detoxification, which increases the effectiveness and prolonging the activity against the insect pests. Did you understand?
Yeah, got it, ma'am.
Okay, ma. And now we are going to talk about the other part. This one is formamidins. Okay. And coming to the formamidins, these are a class of insecticides and acaricides, which are primarily used in agriculture and veterinary medicine. And they are effective against a variety of pests, including mites, ticks, and lice. And these are known for their ability to disrupt the nervous system of the pests, which leads to their control and finally the removal or the elimination. Coming to the structure of formamidin, the example of the most commonly used formamidin is amitraz. This is the structure. If you could see the structure, it has the methyl groups, it has the nitrogen, it has the hydrogen. And if you could see here, the molecular weight of this amitraz is 293.41. And coming to the toxicity of this formamidin. These are highly effective against mites, ticks, and lice by blotting or by damaging the central nervous system. And they have a high margin of safety for most non-target species when used directly. And coming to the toxicity to non-target species, those are beneficial organisms. These show generally low toxicity to humans and domestic animals when used as directed according to the concentration. But if it gets in higher concentration, that leads to nausea, headache, and irritation. Coming to the aquatic life, the formamidins can be toxic to aquatic organisms. So we have to use according to the concentrations given in order to prevent the contamination of water bodies. And it can be harmful to bees and other beneficial insects. Therefore, their use is carefully regulated in many regions. Coming to the mode of action, these formamidins involves interference with the nervous system to target the pest. This is what we already studied maximum in all the cases. And here they act primarily through agonism of autopamine receptors, which leads to the disruption of the nervous system and death in target pests. Okay, how exactly it works? The formamidins primarily acts as agonists of autopamine receptors in arthropods. And this autopamine is a neurotransmitter in invertebrates, which plays a role similar to norepinephrine in vertebrates. Next, disruption in nervous system function. By binding to autopamine receptors, these formamidins cause overstimulation of the nervous system, which leads to increased neuronal activity, which can cause hyperexcitation, paralysis, and death of the pest. This is what about your formamidin. If you could see the additional mechanisms, they may also inhibit monoamine oxidase, which an enzyme responsible for breaking down neurotransmitters, further contributing to their neurotoxic effects. Coming to the things what we have studied under this formamidins, these are insecticides which are used to control the pests. And if you could see the structure, which contains otor formamidin group, the example what we have studied here is amitraz. And then if you could see the toxicity, it shows its effectiveness against target pests with generally a high margin of safety for non-target pests when used appropriately. Potential toxicity to aquatic life and beneficial insects, which are required for careful management. And coming to the mode of action, under this we have studied this formamidin acts primarily through agonism of autopamine receptors, which leads to the disruption in nervous system and finally eventually leads to the death of the organism. This is what we covered under formamidin. And the next one we have to study here is indoxacar. This is another broad spectrum insecticide, which is effective against a number of lepidopteran pests. Despite the carbendin, this substance is not related to the carbamate insecticide. And once this is absorbed or ingested, feeding cessation occurs generally within 2 to 8 hours of time. Okay, and coming to the structure, this is the structure of indoxacar. It has carbon atoms, hydrogen, fluorine, nitrogen, and oxygen. And coming to the mode of action of indexotard, here this indexotard exerts its insecticidal action using sodium channel blockade, disruption of nervous system. The first one is sodium channel blockade, where it is a pro-insecticide. That means it requires metabolic activation to become toxic. Once ingested by the insect, this indexotard is bioactivated to its active metabolite, that is DCJW. This is the N-D-carbomethyloxylated form. This blocks voltage-gated sodium channels in the nervous system of insects. We already studied about voltage-gated sodium channels. These are present in the neurons and muscle cells, and these channels open in response to depolarization of the membrane potential that allows sodium ions to flow into the cell, that initiates the action potential. This is what we have studied under voltage gated sodium channels. And this indexotard, after it gets activated, it blocks the voltage gated sodium channels, which is present on the neurons. And the disruption of nervous system function. These sodium channels, which are essential for the initiation and propagation of action potentials in the nerve cells, by blocking these channels, the indexotard disrupts the flow of sodium ions, leading to the failure of signaling. This results in paralysis and the death of the insects. Coming to the selective toxicity, the specificity of indexotard for insect sodium channels combined with its pro-insecticide nature contributes to its selective toxicity. And it also targets non-target organisms, that is the beneficial organisms, including humans, where it metabolizes indexotard differently, which results in lower toxicity. Here we have covered a few important things under indexotard, where we have studied the importance of the indexotard, which is a oxidizing pesticide, which is used to control the pests. And we have studied the structure of this, and we have studied the mode of action, where the indexotard blocks the voltage-gated sodium channel, which is present in the central nervous system of the insects. After it gets converted into active metabolite, which leads to the paralysis and death of the organism by damaging the nerve signal. This is about your indexotard. And coming to the next part of the molecule is chlorphenapir. It is a pesticide which belongs to pyrrole class of chemicals. That means simply we can call it as a pyrrole insecticide and acaricide. It is used in controlling wide range of insect pests, which are particularly used in agriculture and urban pest management. And this was isolated from the bacterium Streptomyces, and it shows antibacterial activity and insecticidal activity along with antifungal activity. And it is effective against insects, termites, and cockroaches, etc. And it is unique in its mode of action by targeting the mitochondrial energy production in insects. Till now, we have studied the action of these molecules on the nervous system. But here it is targeting the mitochondrial energy production. that means the respiratory system. Okay. And coming to the structure here, it is made up of carbon, hydrogen, bromine, fluorine, nitrogen, and oxygen, and the molecular weight is 407.62 grams per mole. And if you could see the structure, it has the pyrrole ring, okay, with all the atoms. Coming to the mode of action, we already know that it targets the respiratory system in the insects. This chlorphenapper mode of action involves disrupting the energy production in the mitochondria of insect cells. How exactly this works? This is a pro-insecticide. Once it gets activated, like indexocarb, it is also a pro-insecticide. Once it gets activated, it acts on mitochondria, which disrupts the energy production, that is the ATP production. We know that mitochondria is the powerhouses of the cell. Here, this chlorphenapper, once gets activated, which damages the ATP production in the mitochondria. So when there is no sufficient amount of the energy which is required for the survival of the organism, that leads to the death of the organism. That means you are going to see the depletion in the energy. Okay, that's how it acts. In this, the first one is pro-insecticide activation, because this chlorphenapper is a pro-insecticide. It should be metabolically activated in order to show its toxicity on the target organism. And the activated metabolite or chlorphenapper functions as a mitochondrial uncoupler, which disrupts the proton gradient which occurs across the mitochondrial membrane by allowing protons to pass through the membrane without generating ATP. And disruption, when it is showing its action on the energy production, automatically the energy will be, amount of ATP production gets depleted, that eventually leads to the death of the organism. Coming to the selective toxicity, the specificity of chlorphenapper for insect mitochondrial membranes and its requirement for metabolite activation contributes to its selective toxicity, and it even targets the non-target organisms, including humans, and metabolize and respond to chlorphenapper differently, reducing the risk of toxicity when used properly. This is about chlorphenapper. Under this, we have studied about what exactly it is, what is the structure, and the mode of action. If you could see, this chlorphenapper is a pro-insecticide. It should be activated to show its action on the insects, and it has the pyrrole ring with bromine, chlorophenyl, and carbon and other substituents. And it acts on mitochondrial energy production after it gets activated. When it acts on mitochondrial energy production, it disrupts the ATP, which leads to the death of the organism due to energy depletion. This is what we have studied under chlorphenapper. And coming to the last one under this, molecules with different chemistry, the last one is pymetrozine. This pymetrozine is effective against aphids, whiteflies, etc. And it has a systemic and translaminar activity. This is what we studied for the neonicotinoids and pyrethroids in our last classes. And in some cases, the host plants have major effects on the potency of pymetrozine. And it is designed to control a variety of organisms. If you could see the structure of pymetrozine, it is made up of carbon, hydrogen, and nitrogen and oxygen, whose molecular weight is 217.23 grams per mole. Coming to the mode of action, this pymetrozine has a unique mode of action which is not fully understood, but according to the literature, which availability of the literature, we have gathered some of the information about the mode of action. It acts by targeting the feeding behavior of sap feeding insects, causing them to stop feeding and eventually die from starvation. Here, the first one is feeding inhibition. Second one is neurological disruption. Under the feeding inhibition, this pymetrozine disrupts the feeding process of sap feeding insects like aphids, whiteflies, and plant hoppers. After exposure to pymetrozine, affected insects cease feeding within a few hours. Coming to the action of pymetrozine, this impacts specific neurons involved in regulation of feeding behavior. It is believed to interfere with the chordotonal organs, which are mechanoreceptors in insects, which help them in sense internal and external stimuli, including the detection of plant turgor pressure necessary for feeding. This interference leads to an inability to feed properly, causing the insects to starve. That's how it leads to the death of the insects by stopping or ceasing its feeding. And coming to the selective toxicity, this pymetrozine is highly selective for sap feeding insects and has minimal effects on beneficial insects. This selectivity reduces the impact on non-target organisms, making it suitable for use in IPM strategies where ecological balance is crucial. This pymetrozine although shows low toxicity, it did produce some evidences of tumorigenicity in both rats and mice. This is about the importance of pymetrozine. Here we have covered about the introduction of pymetrozine and the structure of pymetrozine and its mode of action. Coming to the introduction of pymetrozine, it is effective. It is effective against aphids, whiteflies, and plant hoppers. It is used to control sap feeding pests, and if you could see its mode of action, it affects the nerves that controls the salivary pump of sucking pests that prevent the insects from inserting their stylets into the plant tissues, causing an irreversible cessation of feeding within a few hours, which leads to the death of the organism due to starvation conditions. This is all about the things which we have studied under molecules with different chemistry, and by this we have completed unit 10. Do you have any doubts in all the things which we have studied yesterday and today? Hello? Am I audible?
Yes, madam, it
Yes.
is audible.
Okay. Is it clear to you all? Did you all understand the clear picture of the molecules, what we have studied under this unit?
Yes, madam.
Yes,
Yes.
And make sure that in your exam, you are writing the mode of action compulsorily. and along with the introduction. Whenever the question is asked, you have to write the first introduction, whatever may be the question. First introduction to be written in your answer, followed by the question what they have asked. Okay, that too in the form of the bullet points. After you describe that, you try to depict the same in the form of a flowchart so that it will be easy for the examiner to understand what you have written in your description to get more marks. Okay, this is all about your molecules with different chemistry. Okay, is there any doubt till here?
No, madam. So,
Okay, now we'll one minute.
Can we get these notes?
Yes, sure. I'll provide the notes, don't worry. But the thing is, I'm worried about the group because I was not added in that group.
Okay.
That is the
Okay,
reason
we
I
will
asked
inform
you
the
to add.
admin, ma'am. We will inform the
Yes,
admin so that, yeah.
Yes, yes. I don't know about that. I thought you are the admins. Maybe
No.
as a group, you form the WhatsApp group.
Okay.
Okay.
Will you please share
As a group.
your number, ma'am, in the chat
even
box so
we are
that,
also having that MSC group, no? MSC Taha. Yeah, admin, no, you can add, or you can tell to someone else who is there. In MSC group, let ma'am add.
yeah, that can be
Amma,
done.
yesterday I have provided my number even.
Okay, ma'am. Yesterday, yeah, since
If
there
you
was an
want, I'll
issue, I
give
could
you
not join.
again now. You please save my number, and if you have any doubts about anything, you please let me know. You
Yeah,
please save my number
sure.
958
One. Yes, ma'am.
170
Yes.
4244.
Okay, ma'am. Thank you. Thank you so much.
958-170-4244. If it is possible, as somebody told, you just add me in your some MSC group. She's
Yes.
telling you ask. Meanwhile, I'll try to communicate with the admin so that it will be easy for us to share the notes. Okay.
Okay, ma'am. Thank you so much.
So now we have completed with
It's
this.
difficult. It's difficult to remember the structural formula. That's very difficult.
But as you're studying the toxicology, you no need to draw the structure. If you could ignore the structure, but try to remember the molecular formulae.
Yeah,
You
okay,
have,
ma'am.
that is important. Maybe while you're drawing the structures, you may do mistakes. To avoid mistakes, try to remember at least molecular formulae. You will be confused in drawing all the structures, remembering all the structures, but you are well versed in chemistry, organic chemistry, then that would be good. If not, also there's no problem. You no need to worry, but try to remember the molecular formulae. Okay.
Okay, thank you, ma'am.
In maximum of the cases, you're going to see macrocyclic lactones only in this particular unit under unit 10. Macrocyclic lactones, where it is made up of pyrrole ring. Okay. You just remember some of the common words for few of the structures. After providing these notes, you try to write down the things in different columns. Try to write down the similarities, try to write down the dissimilarities so that it will be easy for you to remember to write the same in your exam. Okay.
Yes, sir. If mechanism wise the insecticide can be grouped, I think that will be easy for us to remember.
And that's what. In maximum of the cases, these molecules are showing its effect on the ion gated channels.
Yes.
Okay.
Neurotransmitter.
Especially it is showing its action on the central nervous system. In some of the cases, you even saw the action of these molecules on peripheral nervous system too. Okay. So if you could sit and categorize the things while you are reading, it would be easy. If you study these notes, then that would be very much easy for you to understand up to my knowledge. After reading these notes, you just go through the textbook so that it will be helpful. Okay.
Yeah, except
Hope I made sense.
chlorphenaphyr. Yeah, except chlorphenaphyr, others are acting on nervous system, no ma'am?
Yes, yes. But in some of the cases, you are seeing pro-insecticide activity. It should be activated before it shows its toxicity. Hello? Did you see
Ma'am,
that?
Yes,
ma'am,
ma'am.
can you
Yes.
once again say about that pro-insecticide activity? That pro-insecticide, what does it mean, ma'am, actually?
Pro-insecticide means, as I already told you, um, it is an insecticide, but by itself, if it acts directly, it will not show its toxicity on the targeted organism. Okay?
Okay.
It should be activated with the help of some other chemical molecule or some other chemical using any metabolite or any other chemical. Okay? Or by any enzyme. Okay?
Or
Or
like,
by hydrolysis. So once it gets activated, then only it shows its activity against the targeted organisms or the pests what we are talking now. Okay? Understand?
okay, ma'am. Like ion channels and hydrolysis.
That is
No, no,
totally
no.
different.
Like some
No,
insecticide is
no.
injected into an insect so that that insecticide undergoes some metabolism. The metabolite acts on that, activates this insecticide to for further activation. Is that correct,
See,
ma'am?
yes, yes. Some of the I'll whatever the things what we have discussed till now, like milbemycins and fipronil, formamidins, okay, pymecrozin. See, these are the molecules which directly shows its toxicity on the target organisms. Okay, but indoxacarb and this chlorphenaphyr, these are the insecticides which does not show its action on the target organisms directly, but it needs some help. That means in what way? Either by with the help of enzymes or by chemicals. Okay, it needs some help to get its activation to become a toxic. Okay, then it can inhibit, as you told, sodium channels. Then it can inhibit the sodium channels, and then it can inhibit other ion channels to depolarize it and to disrupt cellular respiration. That's how it leads to the death of the organism. Did you understand?
yes, ma'am. Okay.
Hope I cleared.
Ma'am,
Yeah.
activation should happen inside the body of the organism, no, ma'am? Targeted organism. That
No,
activation should happen.
no. The activation will
Is it
not occur.
outside?
Yes, we know
Yes,
that
ma'am.
the cell membrane has the receptors, right?
Yes, ma'am.
Before it acts on the receptor, it must be activated.
Okay.
Okay.
That's what, ma'am. Is it inside the body of the
I'll tell
organism
you.
or outside?
I'll tell you 1 minute
For it,
here.
Yes,
Yes,
ma'am.
I'll tell you. First, the things will go and bind to the receptor. Understand? The pro-insecticide, whatever is present, okay, it will target the organism. Okay. Once it targets the organism, the activation of this pro-insecticide occur by enzymatic actions or by hydrolysis. Okay. Then it shows its inhibition on sodium channels, etc. That will occur inside. because all these channels are present on the where? On the plasma membrane or the cell membrane. So
Yes.
there only you will see all these things. But the activation is required. The receptor should be activated. Then only the channels will be opened. Then only the channels will be closed. If it gets activated, the channels will be opened. If it is not activated, the channels will be closed. Understand?
Yes, ma'am. Okay, ma'am.
Is it
Thank you.
clear to everyone? If you have any doubts, please ask me. I'll try to explain the things. Please concentrate on the dated channels, what we have discussed till now. That is, we discussed about voltage rated sodium channels, and we discussed about calcium channels, and we discussed about some of the ligand rated channels where, 1 minute, where we discussed about, yesterday we discussed about nitrotinate acetylcholine receptors. We discussed about gamma aminobutyric acid receptors. If you could able to understand these receptors, it will be easy for you to write the mechanism or the mode of action of these molecules on these to inhibit the action of these channels, and to how these things protects the pests and all. If you want, I will explain in 5 minutes about this. Coming to the what first one is, we discussed about sodium channels. Okay, we see these sodium channels and the calcium channels
Ma'am, are
are
there any images?
Amma, I don't have much just time.
Oh,
No,
okay,
I don't
okay,
have.
okay.
As you have asked, just I'm telling. If you want, I'll provide the notes for that. I didn't prepare anything for that. It's a general information. Now also I'm reiterating. And now coming to the sodium channels and the potassium, sodium and calcium channels we studied. These are present on the nerve cells, that is the neurons, and also present on the muscle cells. Okay. And these channels open in response to membrane potential that allows the flow of the sodium channels, that allows the flow of calcium channels, so that it will initiate the action potential. That means it plays a major important role in neurotransmitter release and muscle contraction. If there is any alteration in these channels, that means if there is any problem in the flow of the sodium and the calcium channels, that leads to the improper physiological activity of the cell, such that it may lead to the death of the organism. Eventually, it leads to the death of the organism. And coming to the ligand rated channels, voltage rated means that is for the sodium and calcium channels. And coming to the ligand rated channels, yesterday we have discussed about nitrotinate acetylcholine receptor and GABA receptor, where we have seen these things in the central nervous system, and these channels open when the ligand, that is acetylcholine, comes and binds to the acetylcholine receptors. That at that time it gets active, the channel gets activated, and it will open the sodium and potassium ions flow through. That will helps in the neurotransmission. In the same way, the GABA receptor, that is GABA means gamma aminobutyric acid receptor. This is also present in the neurons, that is usually it is present in the brain, where you are going to see the flow of chloride channels here, chloride ions here, sorry, which leads to hyperpolarization of the cell membrane. That's how it inhibits the neuronal activity. So This is all about the voltage rated and ligand rated channels. These helps in nerve signal transmission, and these helps in muscle contraction, and this helps in homeostasis of the cell. This is very important. These channels maintains the ion gradients, which helps in proper cellular functioning. If there is no proper cellular functioning, if there is no proper signaling occurs in the cell to perform its cellular activities, so the cell will be misleaded. It will not perform the actual functions what to do. Because of that improper functioning, the organism couldn't produce its required products for its survival. So ultimately, it leads to the death of the organism. This is all about your channels where these molecules are acting on. Did you understand?
Yes, ma'am. Yes.
Yes,
Hope it is clear, right? Hello?
Yes,
Pretty clear, ma'am. Pretty clear.
Okay, thank you. And now it's already okay. We are going to start our next topic in our next class. If I start, I could able to complete only the introduction, where we have completed only the 10th unit, molecules with different chemistry now. And now we are, and in our next class, we'll try to complete the next part, unit 11. Maybe it is about the insect control agents. If I open also, okay. In our next class, we'll start in a fresh way in about the insect control agents, where we are going to study about what are the synthetic insects, okay, and how these insects, what are the insects and how these insect control agents acts on the insects, okay, how it controls the insects, what is its action, what are the inhibitors, what are the chemical molecules which acts as insecticides. We are going to talk about all these things. Till now we have studied maximum about the natural insecticides, and now we are going to talk about synthetic insecticides. Okay, this is all about your today's topic. If you have hope everything is clear, I'll try to share this notes if you could add me in your group. Meanwhile, I'll also communicate with the admin for the same to share the notes as early as possible. Clear?
Yes, madam. Thank you.
Okay, ma. Shall we leave for the day?
Yes, madam.
Amma, I'll give you the important previous questions also for your reference. I'll prepare and I'll give you. I didn't prepare actually. I was concentrating on your class. Anyhow, I'm preparing the notes accordingly for that particular unit, for that particular block. I'll try to give you the important questions also. Okay?
Okay, madam.
So much, ma'am. Thank you so much.
Thank you.
Thank you, ma.
Thank
Okay.
you so much, ma'am.
Thank you.
Thank you.
Thank you, ma'am. Thank you.
Good day. Thank you. Bye.